Okay, thank you very much, and thank you for the invitation and the opportunity to present to you to our work. We're at an inflection point here. We're very excited about it. I'd like to share that with you. I'd also like to thank Parent Project for the invitation. This is an extraordinary organization. I know that 60 new families are here. I also know this is not a place you want it to be. But I think after being here, I think you realize it is a place you want to be now. And so this is a time of great advancements in the whole field. And I'll share some of what we're doing now. So I'm going to talk about TVN 102, recombinant biglycan as a therapeutic for all forms of Duchenne muscular dystrophy. And TVN 102 is the name I'll use throughout. It's a recombinant form of an extracellular matrix protein called biglycan. So TVN 102 is a bit easier to say than biglycan, and our fervent hope is that next year we'll have a real jawbreaker of a name as we move into IND in the clinic. And so TVN 102 will pass into history. So let me give you an overview of Tivorsan's mission and business just by way of disclosure. I'm a founder and a shareholder of Tivorsan Pharmaceuticals. So our mission is to develop and commercialize an effective and unique biopharmaceutical. In other words, this is a protein, TVN 102, recombinant biglycan, to treat DMD. There are also other indications, but our lead indication by far is Duchenne. A little bit about the company. We're a rare disease company based in Providence, Rhode Island. We spun out in 2010. You can read the rest. We've raised about $14 million, but I think what's very important, key actually, is the logos at the bottom, because this is the organizations who have supported us to this point. Parent Project Muscular Dystrophy has been there from the beginning in our inflection from a basic science observation to a translational program and now into a clinical program. MDA, Charlie's Fund, Nash Avery, NIH has been extremely supportive as well. So that is key, and the partnership is needed to bring this forward. So biglycan, or TVN 102 in DMD. So it would be predicted to be effective in 100% of boys, and I'll tell you why in just a moment. It's not limited by the dystrophin mutation type. Very importantly, it addresses the primary pathological defect in DMD, which is the destabilization of the muscle cell membrane that results from the loss of dystrophin. There's unquestionably a lot of pathology and damage that occurs secondarily to the first insult to the muscle. But what you'd like to do, you very much want to do in Duchenne, is to get at the primary cause and fix that, and then get in early to really slow down the disease at the root of it. And the way to do that is unique in regulating utrophin. You heard some about that yesterday, as well as a very important enzyme. We'll spend a little time talking about NNOS. And it directs the assembly of a functional utrophin complex. Systemic dosing reaches both heart muscle tissue as well as skeletal muscle tissue. I'll talk almost always today about skeletal muscle, but there are some very encouraging signals in heart as well. And also importantly, it's likely that in Duchenne this will be a combinatorial therapy. And biglycan would be expected to be synergistic with existing and emergent therapies, would work on top of truncated dystrophin produced by exon skipping, for example, and would complement drugs targeting secondary pathologies such as the anti-inflammatories, the anti-fibrotics, and the muscle size enhancers, all of which can play a role. But this would come in earlier in the pathway than that. So what's the rationale for biglycan therapy? And this we let nature teach us, okay? So we know that there is a related protein to dystrophin called utrophin. We all have it. It's present during fetal life, it's present when muscle regenerates, and then it goes away. And then in the normal case, dystrophin will come on board. In a dystrophic case, what happens is utrophin is there, but then will persist to a certain degree into mature animals, but it's not localized or at the proper level to keep the muscle fiber healthy and functioning properly. And so utrophin then would be a target along with NNOS. And we discovered biglycan as the molecule in the body that regulates utrophin. Okay, so this is how utrophin localization is regulated in normal individuals. In the absence of biglycan, utrophin goes down, utrophin goes down, you can put biglycan back. And so that led us to the idea that we could capitalize on that. We say, okay, nature regulates utrophin this way, can we do it? Okay, and the idea there is to make recombinant biglycan, which is an extracellular molecule, so delivery is not an issue, and then ask whether or not it can restore utrophin and also nNOS, and it does. And so that's based in biology. So where does that come when one looks mechanistically at this complex of proteins at the membrane that's compromised in Duchenne muscular dystrophy? So utrophin is well established to be important for membrane stabilization, reducing muscle cell death, and resistance to damage. And that's clear. And biglycan regulates the recruitment of utrophin protein to the membrane. But in addition to that, utrophin by itself will not affect a second component of this complex. This is the syntrophin dystrophin nNOS complex, and that's important for fatigue, improving contractility of muscle, and reduced intracellular damage due to ROS or reactive oxygen species. So both of those, so there's a very large amount of data on utrophin, but let me spend a little bit of time talking about nNOS and what its role could be. So we know a great deal about nNOS in MDX mice, just a couple of highlights. We know that properly localized nNOS, and that's important, is important for muscle contractility, combating fatigue. Restoration of nNOS to the sarcolemma improves function and prevents muscle damage in MDX mice. So you can rescue, to an important degree, MDX mice just by getting nNOS to the membrane. Some recent experiments from the Frainer laboratory. But also important is humans. Okay, so what's the role there? First of all, nNOS is lost completely from the muscle membrane in Duchenne. Unlike utrophin, there is some endogenous compensation. nNOS is gone. That tells you something. Functionally, it's also useful as a biomarker, because when you take a measurement, it's always good if you start with something very low and then look for an increase, and nNOS is good for that. Absence of properly localized nNOS in Becker muscular dystrophy, BMD, results in functional ischemia. We heard a presentation related to that from Ron Victor yesterday. Mislocalized nNOS is associated with more severe phenotypes in Becker patients. And properly localized nNOS is strongly associated with mobility status and muscle function in a large study of non-dystrophin neuromuscular diseases. So this is also, as the hallmarks of an important piece of this puzzle that used to be corrected. So some of the data. So TVN102, recombinant biglycan, recruits utrophin to the muscle cell membrane. It was published just a few years back. This is an example of the histology in a systemically treated mouse with MDX mouse. There's over a twofold increase in the levels of utrophin at the membrane. In addition to utrophin, we also restore key components of the complex of proteins that I showed you earlier, notably gamma-sarcoglycan, alpha-sarcoglycan, beta-2 syntrophin, and nNOS that I talked about earlier. And I didn't mention it previously in an earlier slide, but biglycan is an extracellular scaffolding molecule for this dystrophin complex or utrophin complex. It binds directly to gamma-sarcoglycan and to alpha-sarcoglycan, both of which are only expressed in skeletal muscle and in heart. These are the target tissues, so there's, if you will, there's a built-in specificity potentially for biglycan's action by nature of its binding partners. Then looking at the dose response, TVN102 improves muscle health and upregulates utrophin in MDX mice. So the diaphragm, central nuclei go down. This is with a 2-week dosing. This is true in 2 muscles. It's also the increase in utrophin levels, very importantly, in a dose-dependent way. Now those doses, I'll ask you not to remember them, but just keep that in mind, because one of the advances we've made over the past year is a major advance in our dosing with this molecule. Okay, in terms of muscle health and pathology, this is the diaphragm. the most affected muscle in Duchenne and also in MDX mice. So vehicle on the left, on the top, and RHBGN, recombinant biglycan, normalizes to a great degree the pathology that we see, again, the central nucleon, that's the inflammation that's there. And then very importantly, it improves muscle function. So we do two classes of experiments. One is an acute experiment for a couple of weeks dosing, but then we also dose for 3 months because we want to understand what is the benefit of biglycan on the function of the muscle. And one of the key functions you want to do is prevent damage that occurs in use. Because in Duchenne boys, the muscle is fragile. When it's used, it can be damaged. When it's damaged, it sets off that cascade of negative events we've heard about, eventually leading to the death of the muscle fiber and the loss of muscle. So this is a fundamental fix that you want to address. So in this, we did an earlier experiment that was published, but this is an experiment where we also not only did we use the form of biglycan that will be going to the clinic, but also this is an independent validation. So the T2 biglycan, which is a prototype, to TBN 102, it was produced, we dosed the mice, as we see, it was a dose frequency study, and we performed the dose in the Jackson Research. The eccentric contraction, which is this damage, was performed blinded at Penn, in Beth Barton's group, and then the data was sent to us. It's totally done outside. And what you can see is there's a dose frequency response. We dosed every 3 weeks, every 2 weeks, and every week. The blue line is the weekly dosing. And we see for 65% of wild-type levels of resistance to damage. The dashed lines, the horizontal dashed lines are how much damage there would be in a wild type. So we're beginning to approximate that. So this is a robust effect, and again, independently validated. In addition, the biglycan, when you deliver it systemically, distributes to the target muscles, and the top shows diaphragm, and a single dose persists for up to 2 weeks. That's the very top row. And then looking throughout the body using radio-labeled biglycan, I-125, we can see that it goes to heart, diaphragm, quad, and tibialis, so broadly distributed in the mice. Also, we looked at the therapeutic doses, the efficacious doses, at how well tolerated biglycan is. And we'll emphasize this is at the therapeutic doses. Formal pharmacology is on deck for safety. But very good. We didn't see all these curves look the same, and that's really good. That means the mice are tolerating it well. There was one signal that intrigues us, and we will follow up on, which is that in the heart, there's a decrease in the weight of the heart. Now, in the mice, these 3.5 months old mice have been dosed for 3 months of their time. The mouse heart is already enlarging pathologically. The fact that you get a normalization, a reduction in the weight of the heart, is a signal that we're having a positive benefit on the heart. Much more needs to be done, but that is the direction that you would like to see it going in. And the liver and kidney enzymes are also fine, and the health of the animals, weights, etc., organs are all good. Okay, so what have we been up to? So the key thing, this is a protein, and it is complex as proteins are. And our goal has been to develop a formulation, preparation of this molecule that's safe, that's effective, that has good dosing properties. This turned out to be a lot of work. So the starting material, which is the protein produced in cells, in so-called CHO cells, which is the standard in the field. And then from that to develop the processes both in how one cultures these cells, how one harvests, how one purifies and characterizes to get to the optimized clinical candidate. Now the key thing is that all the key steps in this process, which has taken us about 2 or 3 years, longer than we thought, have validated by tests in vivo, so every step along the way. So we're confident that we are improving our activity. So there's a lot of work involved. But the work was worth it, because you'll recall earlier that when we looked at our dose response curves on the left, both in diaphragm muscles, we would see optimal activity about 10 milligrams per kilogram. But with the purified material, we now, in our optimized processes, we now see optimal activity at 1 milligram per kilogram. and that is stable over at least a 5-fold range of doses. So we've got an improvement tenfold in our dosing by improving and understanding our formulation. This is what we will be taking forward to the clinic, so a much better preparation. But that was worth it. The second thing that was also important. Oops. Sorry, got a delay here. Could you put that back up, please? Is this. So all of the studies that I've shown you so far says were in mice. But what about human cells? Okay, so we cultured human DMD myotubes. Okay, and we treated them with biglycan. And this is an example of an experiment where we have done that. And then what we've asked is, does biglycan upregulate the levels of utrophin in the membrane? And again, this is real estate, location, location, and location. Because neither utrophin nor NOS can work unless they're in the right place. And the right place is at the muscle cell surface, and that's what biglycan does, is to recruit it to the membrane. And so we're looking at membrane fraction. So on the left, you can see along the top is the untreated, 0 TDM102, and in the right 1.4, which is the concentration, nanomolar, very good. We see an increase in utrophin, loading control, equal amounts of membranes. Quantifying this by on the right is a standard curve of recombinant utrophin, so we know exactly how much utrophin is there. A kind gift of Jim Urbaski providing us with that standard. And so that allows us to calculate that there's 1.4 nanomolar, again, very low concentration, will give over a 3-fold increase in utrophin protein. In other experiments, we've seen up to 5-fold increase in utrophin at the membrane. Very importantly, with the knowledge of the absolute levels, you can ask the question, is the level of utrophin we're seeing in the membranes comparable to the level of dystrophin that would one see in a normal muscle? And in fact, it is in the same range. So studies that were published first by Eric Kaufman and by others, we know what that percentage is. We can calculate that from these data, and then we know we're in the same range. So this is very encouraging. Okay, so finally, our progress in our plans. Okay, so to summarize the formulation, TVN102 has been developed and improves dosing tenfold. It's a broad dosing range. It's been produced in bioreactors, and methods for purification scalable and suitable for use in producing clinical material have been developed. Absolutely critical to make this into a drug. Importantly, this optimized version, we see greater than a 3-fold, up to 5-fold increase in utrophin at the membrane in cultured DMD myotubes from patients. So what are we doing now? We're currently completing, goal for this summer, the manufacturing process development. And with that then, our goal is to file an IND and enter the clinic by mid-2017, and our stated goal is to have an IND when we talk to you next year. So thank you very much.